Well, hello, everyone. It looks like we're ready to get going for the day. Thank you all for joining us today for our introduction to Lasergene 11. My name is Stephanie Costello, and I'm the Sales Director for DNASTAR, and I'll be coordinating the webinar today. I am here with Jackie Carville, who's on our marketing team. She's going to be demonstrating our new functionality of Lasergene version 11. We just released version 11 last week, and we're very excited to show you what we've been working on for the last several months. You may have noticed that your phone has been muted. We do encourage you to ask questions along the way. Simply just type in the chat dialog and send the chat to the host. I'll then direct those questions to Jackie to be answered for the whole group. If you do need any assistance during the webinar, you can send a chat message to me, or you can also email webinars@dnastar.com. And with that, I'll turn it over to Jackie.
Awesome. Thanks so much, Steph. So before we get started, I'd like to share with you a brief rundown of what we're going to be talking about today. To start off the webinar, we'll be discussing our brand new Lasergene DNASTAR Cloud, followed up with our other brand new application, MegAlign Pro, and we'll be doing some multiple sequence alignment in MegAlign Pro while on the cloud. Then we'll walk through some different next-gen workflows, such as multi-sample Cnv analysis, metagenomic assembly, 16S alignment, and viral integration analysis. Then I'll go through and talk about some new batch processing functions in our SeqNinja application, and wrap up the webinar with a discussion of Protein 3D and give you a sneak peek into our upcoming NovaFold structure prediction. All right. And so with that, I'm going to open up the DNASTAR Navigator here. On our DNASTAR Navigator, we have this integrated help system where it will answer any questions that you might potentially have on our software. So after going from the DNASTAR Navigator, I would like to talk about our new application, DNASTAR on the cloud. So if I pull that up here. So our powerful Lasergene software has been the most published sequence analysis software in peer-reviewed journals for the last 28 years. And as a company, we at DNASTAR are committed to always being on the forefront of the molecular biology software market. We're continuing this innovation by being the first company with an integrated desktop computer and cloud offering. So the great thing about the cloud is that you can access our DNASTAR software anywhere with an internet connection. And some of the advantages to this include access to powerful hardware that you wouldn't have to purchase yourself, the ability to run multiple jobs simultaneously, the ability to monitor your projects from multiple devices, and easy remote collaboration with different peers. So to access the DNASTAR Cloud, you have the option to either use your own Amazon Cloud credentials as well as DNASTAR credentials as well if you don't have those already set up. Now, our flexible licensing options will allow you to purchase a DNASTAR Cloud virtual desktop to really fit your particular research needs. So if we look here, I've already logged in to my account on the DNASTAR licenses page and then clicked on this DNASTAR tab button right here. So when that'll pop up, it'll show your specific license that you've purchased and give you the option to configure a new virtual desktop. So if we click that here, we can see that there's 3 different sized virtual desktop options. And so this small option right here is great for cloning projects, primer design, Sanger assembly, or sequence alignment. So this is really good for all of your smaller molecular biology tasks that you're trying to do. And if you move up to the medium virtual desktop here, we can see that this is great for reference-guided exomes, transcriptomes, and small genome assemblies. The large virtual desktop is a good option for some of your more complex assembly projects. So on the large desktop, our software can do a whole human genome reference-guided assembly in less than 24 hours. So our sales and tech support staff are extremely helpful in talking through all of these different virtual desktop options. And in addition, you can also look at this DNASTAR Cloud technical specifications page for more information on the specifics of these different desktop sizes. So for this particular case, I have already configured this small virtual desktop right here. And so what I would do is select the small virtual desktop and then click the Start Virtual Desktop button. And I've already loaded this up, but you can see that after you've clicked the Start button and it's loaded, you'll have the option to then connect to your virtual desktop. Now, once you've connected to your virtual desktop, this will open up a whole new tab with access to your DNASTAR Cloud. And up here, you can see the Navigator that we've walked through before, and it gives you can see that it again has these pop-out information tabs on the Navigator. And you can open any app directly from the Navigator. Now, an important thing to remember is when you're done working with your information on the DNASTAR Cloud, you have to go back to the licenses page here and stop your virtual desktop with this button. But that is the overall information on the DNASTAR Cloud.
Yeah, we had one question that came in, Jackie. Someone was wondering how secure their data would be on the DNASTAR Cloud.
That's a really, really great question. So here at DNASTAR, through our development process with the DNASTAR Cloud, we've spoken to many different organizations. And we've really found out through this process that there are two major camps with opinions on having data uploaded onto the cloud. And the first camp really thinks that some places will never be secure enough to hold their data. However, the other camp is moving very, very quickly towards uploading data and onto the cloud and doing cloud computing this way. What I can say is that the Amazon cloud has really strong security credentials, which is why we chose it to host our DNASTAR Lasergene software, and that we at DNASTAR cannot access your data once it's uploaded to the cloud. So really this is completely up to your particular organization to decide on whether cloud computing is going to be a good option for you. Here at DNASTAR, we just wanted to make sure that the customers who do want our software on the cloud and enjoy this cloud computing have access to it. All right, great. Well, if there's no other questions, we're going to move right along in the webinar and discuss multiple sequence alignment with muscle in MegAlign Pro. And I'm going to do this since this MegAlign Pro application is brand new, as well as the DNASTAR Cloud. I'm going to do this right on the cloud here. So I'm going to open up this MegAlign Pro from the Navigator. And we'll give it a second to load up here. Here we go. And this opens up the MegAlign Pro interface. So MegAlign Pro is our latest development in our alignment offerings. Lasergene 11 is our first release of this application, but we will continue putting significant investment into this program. We've designed MegAlign Pro so as to be able to support whole genome alignment coming up in the future. But getting started, we can add in sequences from either the Add Sequences project button right here, or we can do File, Add Sequences. So I'm going to select the button. And then here I have 5 different alcohol dehydrogenase sequences here that I'm going to upload from the MegAlign Pro demo data that comes with your Lasergene 11 license. So these sequences right here are amino acid sequences, but you can import DNA, RNA sequences into MegAlign Pro for alignment as well. Now, one of the most striking things now that we're looking at this view is the bright colors here. And right now the sequences are colored by amino acid chemistry. But we can add or remove these colors by selecting sequences, color sequence by chemistry right here. And so we can see that we've removed the colors. But I'm going to want to keep that on for this particular example. Another way to customize your sequence views is by expanding or retracting any annotations or features that we've uploaded along with these particular sequences that we want to align. And so that's really easy to do by checking this plus button or minus button next to the sequence name. And finally, what I personally think is one of the coolest things about being able to customize your sequence views are these panel splits, which are brand new here in MegAlign Pro. And so you can add in as many different panel splits as you want and scroll through the sequences so you can analyze individual sections of the sequences simultaneously. And these are really easy to get rid of, too. You can just drag this right back to the side here to get rid of these particular panel splits. So now that we've played around with the views a little bit, we're going to go ahead and do our alignment here. So to align using muscle, you can either click this play button right here to quickly align, or you can access it under the Align tab in the menu here. We can also see that under this Align tab, we have this Align with Options here. And this allows us to make changes to any of these default parameters for our particular alignment. So it's best to use these default parameters for our first time aligning. And then if we're unhappy with our particular alignment, we can go back and adjust these parameters to customize it. But I'm going to go ahead and do this alignment using our default settings here. And so we can already see that just by looking at the colors that this is aligned up. So we can see these lines with continuous colors. But another way to analyze the efficacy of our alignment is to look at this consensus match histogram on the bottom here. Now this consensus matched histogram divides the total score for the called consensus character by the number of sequences at that particular position. So the higher this bar and the lighter the shade of the green, as seen in these sections here, the greater the agreement for that particular alignment. And so you can also hover over the bars, as I'm doing here, to see the consensus match percentage here. Now, after doing our alignment and being able to analyze our alignment here within MegAlign Pro, we also have the option to send this alignment straight to MegAlign. You can do that through File, Send to MegAlign. And this will open up our DNASTAR MegAlign application. And as we can see that this retains the alignment that we've originally done in MegAlign Pro. And the great thing here within MegAlign is that we have the option to look at phylogenetic trees. So we can do that, view phylogenetic tree here. And it constructs our phylogenetic tree for this particular alignment. So we can look at the relation and the phylogeny of these different alcohol dehydrogenase amino acid sequences. And in addition, we can use all of the other functionality of MegAlign on this alignment, including additional reports and bootstrapping.
Yeah, someone was asking about the number of sequences that you can actually align in MegAlign Pro. Do you know are there any limitations?
Sure, yeah. MegAlign Pro is very flexible, so there really aren't any limitations on the number of sequences that you can align. It's virtually unlimited.
Okay, thank you.
Great. All right, well, awesome. If there's no other questions, then we're going to go ahead and move off of MegAlign Pro and off of the DNASTAR Cloud and discuss a few of our new next-gen workflows. So if I go back to the Navigator here on the desktop, I'm going to go ahead and open up ArrayStar. And so I've opened up this project already in ArrayStar here. And ArrayStar was initially our gene expression visualization program within Lasergene. However, today ArrayStar can do analysis on microarray projects and RNA-seq data. It has strong SNP analysis tools, and additionally supports ChIP-seq and microRNA data. Essentially, ArrayStar is a really great tool when you'd like to do in-depth analysis on any number of samples or numbers of groups, which also makes it particularly useful for cancer genomics and clinical research studies. So the first workflow I'm going to walk you through today is using ArrayStar to analyze copy number variation in a few mutants for a few key genes. So what I would do is click the Start CNV project here, and it will open up a screen like this. So I've already imported here 4 different E. coli mutant assemblies, and these were 4 different E. coli genomes that were run through DNASTAR's SeqMan engine to create a .assembly package. And I've imported these by selecting this Add SeqMan Engine .assembly folder right here. So we want to analyze the differences in copy number variation between these 4 different mutants. The genes we're interested in for this particular project are the THR, A, B, and C genes, which we'll get to later. But since I've already imported these .assembly projects, I'm going to click Next. And here we're at the Setup Preprocessing step where we can adjust our normalization parameters. There's 2 different normalization methods that we have here in ArrayStar for the CNV project, the RPKCN and ZRPKM. And so you would choose your method for this particular workflow based on how many samples that you have. And since we're only using 4 mutant samples for this particular experiment, we want to choose the RPK-CN normalization method. If you have lots of different samples, you'll probably want to choose the ZRPKM method for the CNV project, as more samples in this case provide a better standard deviation between samples for each exon. In addition, we have different advanced options that do exist here to give you further options to customize and adjust your parameters. But I'm going to leave these at the default settings for this particular workflow. In the setup preprocessing step here, you can also choose what features that you're using. So only the features of the types that you select here will be used for mapping your reads. You can select from any of the features that we have in this menu, as well as manually enter in your own feature types. So the setup preprocessing step takes a couple minutes to do. So I've already done that for us. And so if we move into ArrayStar here, we can see these 4 .assembly files have been imported into our experiment list view after this preprocessing step. So for the CNV workflow, we're particularly interested in the gene table here. So I've clicked on the gene table tab here and opened it up in this particular window. So in this case, we want to add an expression level column to see the total raw count in each of these experiments. We'll click OK. And so now we've opened up these 4 total raw count columns here. And if we sort by the name, we can see our 3 genes of interest that we're looking at in this experiment right here, this THRA, B, and C genes. So I'll select those. And if we just look at the numbers in these raw count columns, we can see that it looks like mutant 1 has 0 copies of these genes of interest, while mutant 2 has 1 copy, mutant 3 has 2 copies, and mutant 4 has about 5 copies, in just examining these numbers from the raw count columns. Now, if we want to visualize these numbers, we can open up the heat map here by selecting Graphs, Heat Map. And we can see that our 3 genes appear in these 3 genes and 3 mutants appear in these columns. And all of the reviews in ArrayStar are interactive, so the genes that we've selected in the gene table are going to be selected in all the different views. So we can see that the different colors in this particular heat map correlate with different expression level values, ranging from blue as the lowest to red then being the highest. So that's about it for our CNV sequence analysis workflow. And from that, I'm going to go ahead and move right along into our next-gen workflow, which is metagenomic assembly and 16S alignment. So we're going to go ahead and move into SeqMan NGen for the rest of our next-gen workflows. So we're going to open that up out of the Navigator here. Here we go. All right. So for some background, SeqMan NGen is DNASTAR's sequence assembly software. And a BMC genomics comparison of assembler performance rated SeqMan NGen as the best overall. So in addition to these published studies on how we stack up to the competition, we rigorously test our assembler internally and keep up to date with current next-gen workflows. For further information, our website has great information on all sorts of benchmarking data for SeqMan NGen. So now that we've opened it up, we can see that if we select Create a New Assembly Project, that our assembler is flexible enough to handle a variety of different workflows, from your next-gen genome and exome assembly projects, as well as RNA-seq, ChIP-seq, and microRNA data. For example, we're going to show in this particular assembly metagenomic assembly with host removal. So workflow assemblies sequences against the host genome and then finds matches between remaining sequences and known bacterial species. So for this particular workflow, we're going to be using sequences from the human gut, and we want to remove the host sequences, which in this case are human, from the sample, so that we can find out what bacterial species are present. And so we're going to select, as I said, this metagenomic 16S rRNA assembly. option in this project type section. We're going to go ahead and choose templated assembly with host removal. But as we can see from this assembly type section, that we also have options to run templated assemblies without the host removal step, as well as de novo assemblies with and without this option. As always, if you have a really good template that you're confident in, which we do in this example, you will want to run a templated assembly as opposed to a de novo one. So we're going to continue moving through, and we'll name our project, which in this case we'll name as Metagenomics. And we'll select a project folder, which I'll just select as the desktop here, and a temporary file location. So now we want to input our host files. And here what we're going to do is add a template for the human genome. So I'll add that folder here, which is this human genome template package. And what's really great is that on the DNASTAR website, we have the option to download many different genome template packages for whatever model organism that you're working with. So we're going to move forward to the next step and input our biome genomes. So what we're going to do here is we're going to add a folder from NCBI that contains a few thousand different bacterial genome species. And here we're going to input our sequence files. So the first thing that we have to do in this step is choose our read technology. And as you can see, DNASTAR supports a wide variety of next-gen sequencing technologies for any of your workflow needs. But in this case, we're going to choose this Illumina greater than 50 nucleotides read technology option. And in this case, we actually have 4 unpaired FASTQ files that I'm going to upload. We can see right here. So I'm just going to select those files and open them up in NGen. And moving forward, we can see our assembly options page. Now, advanced options are available for any of our next-gen sequence assemblies, allowing you to customize your parameters to fit your particular assembly. But in this case, I'm going to leave everything at their default settings. So moving forward, our assembly is now ready to begin. We've set up our entire metagenomic assembly for this project, and we would normally click Assemble to begin our assembly. One other important thing to note on this final NGen page is this option to save your assembly. And the save button is really useful because then you can save the script you've created so that you have documentation of the parameters that you set, or if you want to reuse the script for another different assembly.
Jackie, someone wanted to know what the RAM requirements are for SeqMan engine on a specific assembly. Maybe you could,
Sure, yeah, actually, if you want to open up our DNASTAR website here, we actually have really great technical specs for any of our DNASTAR NGen assemblies here. So if you look at SeqMan NGen, we can see these SeqMan NGen technical requirements here, and there's a specific technical requirement page that talks about all of your different requirements for the different assemblies that you're doing. So that's a really great resource for those types of questions.
Great, thank you.
Wonderful, thanks. So after conducting that particular assembly in SeqMan NGen, I've already brought it up in SeqMan Pro here. I want this one. SeqMan Pro is our module for visualizing the results of our different SeqMan assemblies. And so we can see in this project summary window right here, we can sort by name to see all of the species listed alphabetically. And we can, if we scroll down, we can see all of these human contigs right here. So if we exclude these Homo sapiens samples, the samples match 118 known bacterial genomes that we found. We can also sort by the number of sequences and see the sequence frequency. And then that way we can determine the most common species that was found in this particular metagenomic assembly. So moving on from that, I want to briefly discuss the 16S workflow. which is very, very similar to the metagenomic analysis that I showed, but instead you're just using 16S data instead of our different metagenomic reads that we've done. So if I stay in SeqMan Pro, actually, while not particularly meaningful in the context of metagenomics, you can also select Project Statistics to open up a statistics report that gives you additional statistical information on our assembly. So as I said, while not particularly useful for a metagenomic assembly, this median coverage column here will provide useful information for assemblies using your 16S data. So one other cool feature I wanted to point out with these 16S assemblies and metagenomics is this neat feature in NGen where you have the ability to run your samples as separate projects. So once one project is complete, SeqMan NGen will automatically go on to the next one. And this way you can easily set up, let's say 10 assemblies and then let them run over the weekend. And then you can come back on Monday and see the results of all these different assemblies. So that about wraps up the metagenomics and 16S workflows. And
yeah, we did have one question back in SeqMan Engine. You talked about the genome templates packages. What other template packages do we have?
Great question. Yeah, on the DNASTAR website, we have the option to download many, many different genome template packages for pretty much our classic model organisms. So if you're working with zebrafish or mice or human or Arabidopsis or C. elegans or models like that, we will have a genome template package for you. In addition, if you're looking for a specific genome template package, there's a link on that website page where you can contact us and we can help you out with finding something.
Great, thank you.
Great, thanks. So I'm going to go ahead and move forward with our viral integration analysis workflow. And so again, we're going to be using SeqMan NGen for this next-gen workflow. And in this particular example, we're looking for sites within a host genome where the viral genome has integrated. So after an assembly, we would want to look for various chimeric reads that have both viral and host sequence as part of this particular read. So for this example, we're going to be looking at how the HIV virus might integrate into the human genome. So to get started, we're going to create a new assembly project in NGen and move forward and select viral host integration as our project type. So again, we're going to name our file viral integration and select a project folder to save everything out as. So here we want to input our host files. So again, we really want to add this human genome template package right here. And so we're going to select that folder and move ahead. And here we're going to input our different viral genomes. So here we're going to add this HIV GBK file for this particular step. And again, this should look really familiar. We're going to, again, select our read technology. And in this case, it's this HIV greater than 50 nucleotides again. And for this, we have some paired reads. So we're going to add paired reads from our data set here. We'll select these two FASTQ files and import these. And we're going to leave this pair information set at the default insert size. Click OK. We're going to move forward here. Now in this case, we're going to leave most of our assembly options at their default settings. However, we want to make this particular assembly slightly less stringent so that we have plenty of candidate reads to examine in SeqMan Pro. So we're going to customize this minimum match percentage here to be 75%. Right there. And then we're going to go under Advanced Assembly Options and select the Alignment tab, and then change this minimum aligned length here to 25, and click OK. So after we've done adjusting these particular parameters, we're going to move forward, and again, our assembly is ready to begin here. So as with before, I've already done the assembly and opened it up in SeqMan Pro, and we can see it right here. So here in the project window, we can see that we have all the chromosomes shown here, as well as some unlocated contigs that are shown mapping to various reads. If you look at the sequence number columns, we can see exactly how many hits are on each of the chromosomes. And so what's really nice here, we're in the alignment view for this particular window right here, and it has this nice little navigation feature that we're going to open up by clicking on this compass rose. So if we select next coverage, we can scroll through our alignment and see all the different hits and different reads that we found here. I'm going to go back here to the one that I was on. And once we have a couple reads that we're interested in as potentially being a viral integration site, we're going to untrim the end of this read. And this right here would be the section of the viral sequence. So one option that we can do here is select all of this read right here, including both the viral and the human sequence. And we can do net search and run a blast. Now I've already run a blast for us, since that takes a couple minutes, and I'm going to on this particular read. So I'm going to pull that up right here. And we can see that we have lots of HIV matches from this particular blast search. So this gives us really good evidence that this particular read is one of the chimeric reads that we were looking for when looking for these particular integration sites. That about wraps up our host-viral integration section. And that's the end of our new next-gen workflows for Lasergene 11. And so now we're going to shift gears a little bit and move on to another application that's not exactly related to next-gen workflows. And that's our SeqNinja application here within within Lasergene. So SeqNinja is DNASTAR's application for advanced editing of genome sequences and annotations. And so there's a variety of different templates here in SeqNinja that you can use to really easily modify your files. So some of these new templates in Lasergene 11 include this collect template, which allows you to collect multiple sequence sets into one, the sample option, which allows you to sample the sequences in the input set, and the split function, which can split your sequences into multiple files. Now these new templates can really easily be combined with existing SeqNinja templates to easily create a sequence editing script here. So what I'm going to do is, for this example, we're going to look at the collect sequences function. And so I've opened that up here. And what we want to do here is we have this huge folder with thousands and thousands of different genomes in it. What we want to do is collect all of these Vibrio genomes out of this huge folder with all these other ones. Now what we've done is combined this collect function with the extract function right here. So the output of our collection will be used as input for this extraction. So then after we've collected all of these Vibrio genomes from this file with all of the genomes, we have the option to extract many different features here. So we can look and we can see that we can extract different genes, exons, introns, promoters, etc. So let's say for this example we want to extract the genes. So then we can type in like our particular gene that we're interested in finding in these Vibrio genomes. And then we can really easily combine these SeqNinja functions with other existing SeqNinja functions. So I can easily drag and drop any of these other template functions right into the window here to continue building my script. So let's say I want to translate the output of the extract function here. I can just select this translate sequences function and drag it right into this window. And then the output of this extract function will again serve as the input for the translate function here. And then we will be able to translate our final result. So within SeqNinja, you also have the option to save the scripts that you build, as well as obviously save all of your output files. All right, so that about sums up our section on the new batch processing features in SeqNinja. So now I'm going to go ahead and move along to our Protein 3D application. Now, Protein 3D is the core of DNASTAR's structural biology offering, which integrates both sequence and structure visualization for your macromolecular analysis. So one of the great things about Protein 3D is that it has many different views available for you to use. So right now we have displayed here the structure view for Protein 3D, and there are many different ways to move your structure, such as rotating and zooming. So I can move something around, I can rotate our structure, I can zoom in and out. So it's very, very customizable for being able to visualize your particular protein. The analysis view, as we click on here, we can open it up, is our way within Protein 3D to display the results of various applied prediction methods. Now, one of these prediction methods is our DNASTAR B-cell epitope prediction method. And this particular method here has been improved for this Lasergene 11 release. Now, what I'm going to do is quickly walk you through a short workflow that demonstrates two new improved features in Protein 3D, which are both the surfaces feature and the B-cell epitope prediction method that I just talked about. So what we're going to do in this workflow is we're going to learn how to use the epitope prediction method to view the solvent-accessible surface of the likely epitope in relation to the rest of the protein. So I'm going to go back to the structure view here and look at this first. And what I want to do is create a surface for this entire structure right here. So I'm going to go under the Surfaces panel, right on the side here, under the Explorer menu, and I'm going to create a new surface, which is a high-quality solvent-accessible surface. I'm going to select that there. And if I go into the Style panel, I'll be able to change the color for the current selection. So let's say I want to change this to green and click OK. Oh, it looks like I have a specific region selected. So instead I want to create a surface for the entire protein. Sorry there. And again, I'm going to select the fill color as green. So there we go. I've created a surface for the entire protein and colored it green here. As I said before, the surfaces are greatly improved in Lasergene 11, making them tighter and more biologically accurate. So now if I go over to the analysis view and switch gears a little bit, I'm going to want to open up the Methods panel and select the B-cell epitope prediction method. If I scroll down, I can see that this has appeared in the analysis view, and we can expand this to see these different methods here. So in the analysis view here, we can see both the antigenic regions, which are shown in light pink, and the highly antigenic regions, which are these dark pink models. So we can also expand here to see individual graphs showing confidence scores for these particular regions. So these graphs with the confidence scores indicate the probability that this is indeed a B-cell epitope. So what we can do here is we'll highlight a particular region of interest. So I might be interested in, let's say, this region right here because of the highly antigenic confidence scores in this region. So after I've highlighted that region, what I'm going to want to do is go back to the Explorer panel, and then I'm going to change the color on the surface for this particular selection. So let's say I want to make this selection blue. So I'll select that. And now if I go back to the structure view, I can see that this is the potential region for our epitope. So those are a couple of our new features here in Protein 3D.
Yeah, someone wanted to know what the input file format is for Protein 3D.
Oh, that's a great question. So we can do either protein structure files, which you can download straight from the PDB database. And if you don't have a structure file for your particular protein of interest, you can also use a sequence text file that you can open up. And you can still do the analysis view and do all of the applied methods on that particular sequence, even if you don't have a structure file as visualized here. Great, thanks. So to wrap up then, I want to give you a sneak peek of one of the features that we're currently developing. So DNASTAR NovaFold, right, as we can see in this bottom panel here, is our structure prediction software, which is integrated into Protean 3D. NovaFold is based off of algorithms that have won the CASP structure prediction competition for the past 8 years. And DNASTAR has taken these algorithms and created a great graphical user interface for them. So what you do is you begin in Protean 3D, and your structure prediction is sent up to the cloud and processed, and then returned to Protean 3D for full analysis. Now for Lasergene 11, we are only releasing the preview mode of NovaFold, but we'll have full structure prediction functionality coming soon, later this spring. So NovaFold makes its predictions through a hybrid method which identifies structural fragments from multiple different templates, and then will build the fragments of sequence not matching this particular template. So you can access NovaFold through Protean 3D in a variety of different ways. So you can either access it through the details panel here, or you can access it by selecting structure, predict structures in NovaFold. I already have the tab open right here, so we're going to look at this. So what we can do is we can click on any structure prediction that we've already done, and if we go back to the details panel, we can see all of the details on this particular structure prediction. And the details can give you a really good idea for either how long the prediction might take based on the number of residues in that particular protein, as well as let you know which templates were used for that particular structure prediction. And you can click on this prediction and open it up in our structure and analysis views here. And then you can do any of the different workflows that we've already shown you within Protean 3D. It's highly integrated. So some of the advantages of this particular application are, as I said, its integration into Protean 3D, and your ability to do different structural alignments and other analysis with the structure prediction that we've done. Another advantage here is being able to have the structure prediction conducted on the cloud, so it gives you really high performance for this computationally intense prediction task without actually having to purchase this physical hardware yourself. So as I said, that we have the preview mode now in NovaFold, so with your purchase of Lasergene 11, you'll be able to test out this and new feature that will be coming soon.
Great. Well, it looks like we're all done with the presentation. If we didn't get to your question during this webinar, we will follow up with you via email after the webinar. If you think of other questions later, you can email those to webinars@dnastar.com. I also want to point out that on our website, we do have a really large collection of videos and past webinars that we've hosted. I encourage you to visit our videos page or webinars after and check out all those great videos. We also do plan on hosting additional webinars on some of the next-gen workflows that Jackie showed today. We don't have them scheduled yet, but we do plan on sending out invites, and we'll have topics and dates and times posted on our website soon. If you are interested in trying out a free trial of Lasergene 11, on every page of our website, up in the upper right-hand corner, there's a free trial button. Simply click that, fill it out, and you'll get you set up with the Lasergene 11 free trial. So thank you, everyone, for joining us today, and we hope you found this webinar helpful. Great. Thank you.